The overall goal of this 2D gas chromatography data acquisition method is to characterize the aqueous byproducts obtained from hydrothermal liquefaction, or HTL, of algae.
This method can help answer key questions in the biofuels field, such as identification of the organic species that report to the aqueous phase during hydrothermal liquefaction of algae. The main advantage of this technique is that it improves peak capacity, resolution, and avoids correlation of chemical compounds.
This method is important to generate data for biorefineries, which produce considerable volumes of wastewater that must be either treated or further processed to fuels and chemicals.
This method can provide characterization data that cannot be produced from other analytical techniques such as one-dimensional gas chromatography or liquid chromatography.
We first had the idea for this method when we were analyzing very complex organic mixtures from biofuel production processes.
Use a gas chromatograph equipped with a quad-jet dual-stage cooling-based modulator and time-of-flight mass spectrometer for these experiments. Configure the autosampler to inject 1 microliter of each sample or standard into the gas chromatograph, or GC. Use a randomized block design of sample and standard injections for the autosampler sequence, as described in the literature. Ensure that both edges of both primary and secondary columns are cut straight without sharp edges. Then, connect the primary and secondary columns using a press-tight connector before the modulator. Ensure that the glass liner, non-stick liner O-ring, and septa for the GC injector are new and free of contamination.